Hello and welcome to the Science AAAS and Science Signaling webinar. My name is Annalisa Van Hook and I'm the web editor at Science Signaling. In today's webinar we'll be discussing the role of epigenetics in promoting cancer progression. This webinar is the fourth and final in the series focusing on the signaling pathways that allow cancer development and progression, the emerging research in identifying and targeting these pathways, and innovations in developing new cancer treatment options. Tumors are complex tissues in which the cancer cells evolve traits that promote their own survival and progression of the disease. Some of these traits involve epigenetic mechanisms that affect the expression of genes that in turn stimulate cell proliferation or survival, or allow the tumor cells to evade the immune system. Our speakers today will focus on chromatin modifications that can lead to oncogenesis, and they will address epigenetic changes that have been observed in tumor tissue and outline ways in which epigenetic pathways can be targeted to treat cancer. It's my great pleasure to introduce our guest speakers for this webinar. They're Dr. Stephen Baylin from the Johns Hopkins School of Medicine in Baltimore, Dr. Charles Roberts from the Dana-Farber Cancer Institute in Boston, and Dr. Ali Shilatifard from Northwestern University's Feinberg School of Medicine in Chicago. A very warm welcome to all of you, and thank you for being here with us. Before we start today's webinar, I have some information that our audience might find helpful. Up to the right of your screen, you'll see our speakers. Click on the View Bio link to see more details about each of them. Under the slide window is the Resources tab, where you can find additional information about technologies related to today's discussion, or a link to download a PDF of the slides. Our speakers will give short presentations, and after that we'll have a question and answer session in which the speakers will address some of the questions submitted by you, our live online viewers. So if you're joining us live, start thinking about some questions and submit them at any time by clicking the Ask a Question button below the slide window, typing your question into the box, and then clicking OK. Please remember to keep your questions as short and clear as possible, as that will give them the best chance of being put to our panelists. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant widgets at the top of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology and the Koch Institute for their sponsorship of today's webinar. Now I'd like to introduce our first speaker for this webinar, Dr. Stephen Baylin. Dr. Baylin is the Virginia and D.K. Ludwig Professor of Oncology and Medicine and Chief of the Cancer Biology Division and Associate Director for Research of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins. His research interests include cellular biology and the genetics of cancer, specifically epigenetics and the silencing of tumor suppressor genes and tumor progression. Dr. Baylin has served on the American Association for Cancer Research Board of Directors and chaired the AACR Special Conference on DNA methylation, imprinting, and epigenetics of cancer. Welcome, Dr. Baylin.
Yes, welcome everybody. Today you're going to be hearing from each of the speakers how abnormalities in epigenetics are throughout cancer and the possibilities that you can target these for reversal to reprogram cancer cells. Since I'm the first speaker, I will introduce briefly the concept of the epigenome and epigenetics, so we're all on the same page. The definition of epigenetics can be traced back to the biologist Charles Waddington, who was trying to understand and propose that each cell in an organism could switch its phenotypes, but it would do so without changing the fundamental genetic background of the organism. Fast forwarding to our molecular age, we now define epigenetics as heritable changes in patterns of gene expression and function which create a new phenotype, but without a corresponding change in the DNA sequence. So this is a closely integrated set of events with the genome and essentially serves as the software for driving the hard drive of DNA, which I'll define in a second. The advantage of targeting these abnormalities is that unlike mutations, epigenetic changes can be reversed, both experimentally and in organisms. And this gives us the possibility that by doing so, we can revert their abnormalities to a more normal pattern to reprogram cancer cells. This slide shows the concept that all of the information that constitutes a cellular function is found in the hard drive of DNA. but essentially epigenetics serves as a software package where the round balls shown here of nucleosomes package the DNA in such a way that the function of the individual genes can be called on. When the nucleosomes are in an open pattern, like you see on the left, that usually subserves an active gene at the transcription start site. But they can switch to the most closed pattern in the top right of your slide, where the nucleosomes are closed, and often a change of DNA methylation accompanies that change. And an example of how one can reverse abnormalities is to change a methylated cytosine, which is next to a guanosine, or the CpG nucleotide, back to an unmethylated cytosine, and that can change to re-expression of the gene and bring the gene pattern in a more open and back on. On the current slide, you see a vision of today's epigenome, which is complex, and I will describe it only very briefly. Epigenetics is also about three-dimensional structure. So you see on your slide a point labeled gatekeeper. The gatekeeper can be thought of as the gene start site, and if it is closed by DNA methylation, for example, or nucleosomes, can block the function of the gene. But genes are also talked to, as you see towards the top of your slide, by distal regulatory elements or enhancers, which can be looped in towards the promoter start site of genes to regulate their function. DNA methylation and chromatin, which I'm going to mention briefly, as will the other speakers, regulate both promoter regions and enhancer regions. To the far left of the slide in the orange box, I will remind you and will be talking about the fact that non-coding RNAs or the non-transcribed protein functioning RNAs in our genome are also controlled by epigenetic events and the same types of changes in DNA methylation and chromatin. I will present it as an example today, going back to the gatekeeper, the fact that when you have CpG-rich areas or CpG islands around gene promoters, 90% of the time, those cytosines at that site are unmethylated. But cancers can devise or evolve de novo methylation in those changes, which can silence genes as an alternative to mutations. And in the orange letters to the bottom left of the slide, I show examples of key tumor suppressor genes that can either be mutated or have this DNA methylation change as an alternative way to lose their function. So I'm going to concentrate on that promoter DNA methylation change. And in the next slide, I want to point out that this change and some of the changes that the other speakers will allude to often begin very early in the neoplastic process, often in pre-invasive stages, often in stem cell compartments, before one sees a transition with progression towards cancer, invasive changes, and metastatic changes. And so these abnormalities are progressing throughout cancer. Now, one of the exciting things about the epigenome, and you'll hear more about this from the other speakers, is that many of the mutations that are now being regulated in cancer encode for the genes that control the epigenome, setting up the possibility that those changes in the genetic part of the hard drive the effects can be reversed by targeting downstream abnormalities that they may cause in the epigenome. And all of those mutations, like other driver mutations that you're more familiar with, are also evolving during tumor progression. On this slide, I want to show you a concept about reprogramming the cancer epigenome, such as changing DNA methylation. Hundreds of genes in every individual patient's cancer have those gatekeeper-type methylation changes that can arise. And I show in the asterisks around this famous Weinberg-Hanahan wheel of pathways that can be abnormal and drive cancer, that genes affected by that change are throughout every one of those pathways. This sets up the possibility that by targeting and reversing those abnormalities, one could revert simultaneously many abnormal pathways back to more normal ones. As shown by the circle that is going to arise around immune activating pathways. Let's see if that will come up. Just wait 1 second. Just 1 second. So what you should be seeing now is the possibility, as I mentioned, that if you reverse the abnormalities in multiple pathways at one time, it raises the possibility that you could reprogram the cancer cell to a more normal state. So if you're on the slide with the round Weinberg-Hanahan circles, if we hold there, I've drawn a red line around immune properties, and I'm going to illustrate to you that one thing that can be reversed in tumor cells by reverting to a more DNA methylation normal pattern is to help the tumor reactivate its immune attraction systems. Now, what you should see on your slide, I'm going to give you an example of an epigenetic therapy trial to reverse abnormal promoter DNA methylation in advanced non-small cell lung cancer. And I'm going to illustrate three possibilities that we've seen early in this clinical trial, and I'm going to discuss several of them. Of 65 initial patients given a therapy that I will describe for you in a moment, 2 of those 65 had complete regressions of this very advanced and lethal tumor just with the therapy alone, which I will define for you. 25% raised the possibility that using epigenetic therapy prior to subsequent therapies may be a way to sensitize patients to things like chemotherapy, and I'm going to show you the possibility that it may do so for immunotherapy as well. And all of these that I'm mentioning to you are in larger trials, which are now in progress, although we're going to have to wait the outcome to know whether these hypotheses are all validated. In the next slide, I show you a patient receiving a low dose of a DNA demethylating agent called Vidasa, plus an inhibitor of histone deacetylation, which converts genes to a more active mark. And if you look at the red arrows to the top left of each of the panels, you'll see the lesions of lung cancer in this patient. If you focus on the liver, you'll see over several months of therapy the disappearance of that metastatic lesion along with the chest lesions disappearing. And this was a very durable response that lasted for several years in the 2 patients that had this change. The challenge now is to personalize such therapies by recognizing exactly what epigenetic changes accompanied all this that would predict the response in such patients. The next slide is going to show you a waterfall plot. And it is in the same trial of 65 patients and shows you that in these 65 patients, 29% of them, after they progressed on the epigenetic therapy, went on to have RECIST criteria responses to a subsequent chemotherapy. And this is another hypothesis, an early signal that you can use epigenetic therapy to sensitize and revert pathways such that the patient's tumor may be more sensitive to chemotherapy. The next slide is going to show you an early example in the same 65 patient trial that patients who received epigenetic therapy first, after they progressed, went on to subsequent immune checkpoint therapy, which I'm going to define for you in a moment. A very, very topical and promising form of therapy now. In the bottom left, you see one of these patients. You'll see by the double arrows the lesions in the lung after they came off the epigenetic therapy, and then fast forward 21 months into the immune checkpoint therapy, showing you that those lesions have regressed. The 5 patients who showed these kinds of changes are shown in panel B at the bottom. 2 of those 5 did go on to progress, but only after a 24-week period that only 20% of patients on the immune checkpoint therapy alone pass, and the other 3 patients in the downward curving lines all had virtually complete responses which have lasted for several years. And this is a concept which is now in clinical trials to validate this possibility. The next slide defines immune checkpoint therapy. And basically, if you go to the top right of the 4 panels, you'll see that what immune checkpoint therapy does is to break an interaction between ligands on the tumor cell, like PD-L1, and receptors on T cells, like PD-1, wherein the chronic interaction of those 2 events renders the T cell immunologically inactive or tolerant. What the immune checkpoint therapy does is use antibodies to either the ligand or receptor, like anti-PD-1, which was in those trials for the lung cancer patients, and breaks that tolerance such that the T cell now becomes again an effective immune cell. And what we have done in our laboratory with colleagues is to get a signature for cancer cells responding to low doses of the DNA demethylating agent used, 5-ASA or Vidasa, by exposing cells for a transient 3-day period, then taking the drug away, resting the cells, looking how they behaved in animals for self-renewal properties, but also during the rest period, as shown to the far right of your slide, getting genome-wide DNA methylation patterns, genome expression patterns at the RNA and protein levels, and doing proteomics. And with a battery of those signatures, one can look at primary tumors for the baseline levels of those signatures as a predictor of how they might respond to the drug. And I'll use those signatures in the next slide to show you that there is a 300-gene signature called ASA-induced immune genes, which we call AIMs, which are all immune pathway genes that are upregulated by the DNA demethylating drug Vidasa. And in these pathways, upregulated are antigens on the surface of the tumor cells, MHC1 complex proteins for processing those antigens, and a complex immune attracting signature, which I'm going to show you we called out of this 300 genes to a 20-gene panel shown in the next slide, which shows that this drug upregulates in the tumor cells essentially a viral defense pathway. It's virtually like making the cells simulate that they have seen a viral infection, and this is at the double-stranded RNA level. So azacitidine upregulates this entire pathway, which is at the end of the day, by shown in the bottom circle, an interferon beta stimulating set of events that can attract immune T cells. And we've blocked each of the steps in this viral defense pathway, as shown by the X's here, to prove that blocking them inhibits the drug from activating the pathway. Now, in the circle that's going to come up, you'll see that it goes around what are called ERVs. And ERVs are endogenous retroviral sequences that are in our genomes. And this drug activates transcripts from those ERVs. And that is one of the driving events in this viral mimicking type of scenario, which drives the viral defense pathway. And I'll come back in the end slide and show the model here again. So you drive up the ERVs, and you drive this viral defense pathway. I will remind you in the next slide that, go one more, that ERVs have been in our genomes for millions of years. They constitute some 8% of the genome, retroviral elements, and they are an example of how DNA methylation and chromatin events can silence the sequences, because the ERVs are silenced in most normal, mature cells that can be demethylated by the drug when these tumor cells have methylation at the start sites. So my last slide will show you the paradigm, and I will carry you through it very briefly. On the left side is a tumor cell. DNA MTIs at the bottom means that one treats that tumor cell or treats the patient with the tumor cells and activates this complex immune signature for presentation of antigens on the surface and upregulation of the proteins, the MHC1 class 1 proteins that process them, upregulate this viral defense pathway, stimulate an interferon response, which in turn can attract T cells. Now, at the time of that attraction, the tumor cells will have PD-L1 on the surface, and the T cells will have PD-1, so there may be a situation of tolerance. But when you come in, subsequent to the DNA methyltransferase inhibitor with a anti-PD-1 antibody, one can break that tolerance and potentially reverse the T cell inactivity to make them active T cells. So again, in final, clinical trials are up for this using an epigenetic therapy, reversion of DNA methylation, to activate the immune processes and attraction in the tumor cells to bring in T cells and then render those T cells active. And I thank you for your attention. Apologize for the slides.
Thank you, Dr. Baylin. We'll move along now to our next speaker, Dr. Charles Roberts. Dr. Roberts is an associate professor in pediatric oncology at the Dana-Farber Cancer Institute and Harvard Medical School and deputy chief scientific officer of Dana-Farber Cancer Institute. Dr. Roberts also serves as Scientific Director of the Pediatric Solid Tumor Program and co-chair of the Pediatric Institutional Review Board at Dana-Farber. He's been elected to the Society of Pediatric Research and the American Society for Clinical Investigation. His laboratory studies the role of the switch-SNF chromatin remodeling complex in driving cancer, and Dr. Roberts has recently accepted the positions of Cancer Center Director and Executive Vice President at St. Jude Children's Research Hospital and will be moving his laboratory there shortly. Welcome, Dr. Roberts.
Thank you. So my laboratory is really interested in understanding the interrelationships between chromatin remodeling, transcriptional or epigenetic regulation, and oncogenesis. And in particular, we're interested in the switch-SNF or BAF chromatin remodeling complex. We first became interested in this a number of years ago when I was doing my postdoc, when Olivier Delatre's group reported in Nature that there were biallelic inactivating mutations in a subunit of this complex present in a very nasty type of pediatric cancer called malignant rhabdoid tumor. It caught our attention for two reasons. First, this represented the first link between ATP-dependent chromatin remodeling and cancer, so therefore potentially new ways to understand oncogenesis, also potential new ways for insights into therapy. But it was also of interest because history has taught us that genes that are mutated in the earliest onset pediatric cancers often play a fundamental role more broadly in cancer. So for instance, retinoblastoma is a rare pediatric cancer, but it's the disease that taught us about tumor suppressors and the importance of the RB pathway. So the disease that we began studying as a model or canary in the coal mine, rhabdoid tumors, highly aggressive cancer of early childhood that occurs in kidney, brain, and soft tissues. They're quite lethal, and the majority of kids die of their disease within a year of diagnosis. And ultimately, there are biallelic mutations in this gene that goes by 4 different names, including SNF5 and SMARCB1, present in nearly all cases. Now, the switch-SNF chromatin remodeling complex is highly evolutionarily conserved. Shown here are two flavors of the mammalian complex at the top, the BAF and PBAF. In green are the ubiquitous or core components that are expressed in all cells, but you'll see that there are a number of lineage-specific subunits encoded by multi-gene families. And so Jerry Crabtree has argued there may be several hundred variants of this complex that can help enforce lineage specificity. What we know about the biochemistry of the complex comes from people listed on the left and others, but bottom line, in vitro, the complex can bind to nucleosomes and slide them at promoters and enhancers, but what it's doing in the setting of three-dimensional chromatin structure is in the cell's nucleus has been much less clear. So we wanted to understand, was this really a tumor suppressor, and could we make a model? And so here's what happens with conditionally targeted mice. If you turn off SNF5, 100% of the mice develop cancer, and the median onset is only 11 weeks. This is remarkably rapid for turning off a single gene. In comparison, it's twice as fast as p53 inactivation and even faster than the combination of p53 deficiency with RB haploinsufficiency. So it suggested a potent role in tumor suppression. We began to then use the model to study the biology. You remember that one of the questions we were interested in was this revealing a broader role in cancer? As we initially began working on it for the first few years, that wasn't clear. And then in 2010, with publications in Science in the New England Journal, the floodgates opened. And it's now become clear that at least 9 subunits of this complex are recurrently mutated in cancer, collectively now exceeding 20% of all cancers in a nice paper from Sagal Kadosh and Jerry Crabtree. Thus, the switch-SNF complex has emerged as a major tumor suppressor complex, but also one that is only recently recognized to have such a broad role in cancer. So one of the first questions that we began to ask is, what is the primary mechanism of rapid cancer onset following SNF5 loss? Is it genetic or epigenetic in nature? And what I mean by that is SNF5 is genetically lost, but what are the downstream effects that drive cancer? The complex is involved in transcriptional regulation, but it's also been implicated potentially in DNA repair. So we looked and because it's published, I'll just summarize. We asked the question, does SNF5 loss lead to defective DNA repair or genomic instability? We were unable to detect a substantial role in DNA repair, and when we looked at the cancers, they were diploid. So these aggressive cancers were diploid, they were not aneuploid. And when we sequenced them and looked at the exomes, what we found was that they had the lowest somatic mutation rates ever detected across cancers that had been sequenced at the Broad Institute. On the right are the cancers that I learned about in graduate school, lung cancers and melanomas. All the way on the left are the rhabdoid tumors, which in this logarithmic plot were more than a log-fold lower. And in fact, in the exomes, when we looked, some of the tumors had only mutations in SNF5 itself. So this suggested to us then that oncogenesis in the absence of SNF5 appears to be transcriptionally based. It's not causing aneuploidy or scrambling the genome or causing high mutation rates, despite the fact it's driving extremely rapid cancer onset. So that leads to the question then, what are the chromatin mechanisms that underlie oncogenesis driven by SNF5 inactivation? One of the things that we began to wonder about was data that had come out of Drosophila, where Drosophila geneticists interested in the polycomb pathways asked what can suppress polycomb and found that mutations of switch SNF did that. And so we moved forward in asking, is this relevant to mammalian biology? Is it relevant to cancer? And I'll just summarize that we found epigenetic antagonism between SNF5 and EZH2, a polycomb 2 repressor complex member, such that they were antagonistic effects at polycomb targets. SNF5 inactivation led to increased levels of EZH2 and H3K27 trimethylation at polycomb targets, and this led to silencing of the polycomb targets. We asked, is this relevant to cancer formation using genetically engineered mouse models? We got an EZH2 conditional mouse from Stu Orkin's lab. In the red is, again, the SNF5 cancer onset. And when we add in EZH2 conditional allele, it now completely blocks the cancer formation. All of the cells of origin are still there. They simply don't transform. It's important to point out that this is a specific effect because if we inactivate EZH2 on a number of other cancer backgrounds that have high levels of EZH2 but are not driven by switch SNF, we do not see any effect on tumor onset. And so the model then became of interaction in gene regulation between switch SNF and polycomb to regulate polycomb targets and lineage specification, such that mutation of SNF5 perturbs this balance, leads to dysregulation of polycomb targets and a loss of control of lineage specification and differentiation, which contributes to oncogenic transformation. That then prompts the question, is EZH2 dependency in SNF5 mutant cancers relevant to human patients? And so here, Epizyme has gone forward with a clinical trial. I do not have a relationship with Epizyme. And they've launched a phase 1, and because of our data, we're able to enroll a patient with recurrent metastatic malignant rhabdoid tumor. And it's been one patient they've enrolled, and that patient has got a complete response, shown in the top in the PET scan and also in the CT scans. And as I was last aware, in June, now 10 months in, the patient has a sustained complete response to single-agent therapy. They've gone on to enroll additional patients and are now have announced that they're planning a specific trial for SNF5 deficient cancers based upon efficacy that they're seeing. So in addition to pursuing hypothesis-driven mechanisms, and here I've only talked about one of the chromatin pathways that we've looked at, we've also gone at less biased ways to look for vulnerabilities that are caused by mutation of the switch SNF complex. And so to interpret these, I first want to make one point, and that is, what is switch SNF without SNF5? Well, the loss of SNF5 does not cause the complex to be inactive, but rather it leads to an aberrant residual complex that drives cancer onset, and we found that's true of the other subunit mutations as well. So we then went forward with something called Project Achilles in collaboration with Bill Hahn's laboratory, which is essentially a near-genome-wide screen, initially of 50,000 shRNAs against 11,000 genes. It's now up to 19,000 across several hundred cancer cell lines. Because of the frequency of switch SNF mutations, there were enough cell lines already that had BRG1 or ARID1A mutations that we could look at. What we found was that ARID1B was the #1 dependency in ARID1A mutant cancer cells, so another subunit of the switch-NIF complex when the mutation of ARID1A. When the BRG1 subunit is mutated, again we find another subunit of the complex that is the #1 vulnerability, and that is BRAMA. And so essentially the theme that has been emerging is that residual switch-NIF complexes can constitute a vulnerability in cancers that contain a switch-NIF subunit mutation. Mutation of BRG1 or SMARCA4 results in dependence upon BRAMA or SMARCA2. ARID1A mutation leads to dependence upon ARID1B. And the question then going forward is, and we're interested in this, mutation of the other tumor suppressor subunits, do we get, again, specific dependencies? So as I then summarize what the model is in thinking about these things from a therapeutic standpoint, one way to go at them, because these are tumor suppressors that genes are deleted and lost, you can't directly target them, is to look at downstream targets, and indeed there's a CDK4 inhibitor clinical trial based upon cyclin D1 dependency. That's going on, but it's one of the things that makes me nervous is that this is essentially targeting a single pathway, and we know that these remodelers regulate thousands of targets. If we move up to the level of chromatin, we look at now the EZH2 inhibitor clinical trial, and here we think we're targeting multiple pathways based upon the chromatin regulation that's the fundamental biochemistry of the complex, and time will tell if those responses are even broader. And then we move to the question, can we directly target the residual complex itself? And now we and a number of pharmaceutical companies are pursuing this as well. So in conclusion, inactivating mutations of genes of the switch-NIF or BAF chromatin remodeling complex frequently occur in a variety of cancers, both pediatric and adult. SNF5 deficient human cancers contain remarkably few genetic mutations, suggesting an epigenetic mechanism by which SNF5 loss promotes cancer. Inactivating mutations in switch-NIF subunits may create specific dependencies upon other subunits of the complex. And finally, that mechanistically based targeting of chromatin structure and epigenetic regulation may hold therapeutic promise. This complex has now emerged as being broadly relevant to human cancer, and these now, I think, constitute some of the most exciting ways to begin to look at this, and we're now beginning to get early data. So I will end there, and back to you, Annalisa.
Okay. All right. Thank you, Dr. Roberts. Our next speaker today is Dr. Ali Shilatifard. Dr. Shilatifard is the Robert Francis Furchtgott Professor and Chairman of the Department of Biochemistry and Molecular Genetics at the Northwestern University Feinberg School of Medicine in Chicago. Dr. Shilatifard is a Deputy Editor at Science Advances, a member of the Senior Editorial Board of Science, and serves on the Scientific Advisory Boards of Genentech, Keystone Symposia, and the Max Planck Society. His laboratory studies the protein complexes that regulate gene expression during normal development and how mutations that affect the components of these complexes are associated with human cancer, focusing specifically on childhood leukemia. Welcome, Dr. Shilatifard.
Thank you, Annalise. Good morning, everyone. If it's morning or good afternoon for some in the East Coast and West Coast or in Europe, good night. So my laboratory, as Annalise mentioned, is in the Department of Biochemistry and Molecular Genetics at Northwestern University in Chicago. And for decades, we've been very much interested in defining how chromosomal translocation is involved in pathogenesis of hematological malignancies. And just following this process of chromosomal translocation and the role of these translocation partners in leukemia has brought us into the area of chromatin biology and how chromatin is involved in this process. So as shown in this slide, there we go, American Cancer Society published this fantastic story many years ago indicating by the time that most of us turn about 60 years of age, the chances of getting cancer for us is about tenfold higher than we were in the early 20s. And for decades, we have been studying the role of factors that involve in cancer pathogenesis as a field. And what we have learned is that tumor suppressors and oncogenes are central in cancer pathogenesis. But knowing this fact of the role of tumor suppressors and oncogenes has not really resulted in defining and developing targeted therapeutics for treatment of cancer. And so what we have learned is that large number of factors within our genome, which are called protein coding genes, are involved in regulation of gene expression and cellular development. And for over a decade, as a community, we have been sequencing the protein coding genes in trying to understand what's the role of these factors in development and how the misregulation is involved in pathogenesis of cancer. Protein coding genes encode about 2% of our genome. The other 98% of the genome, which initially was considered to be junk DNA, and now we refer to as regulatory elements, were so ignored. And what we have learned during the past few years is that enhancers or regulatory factors that are involved in regulation of gene expression are the tip of the iceberg for defining cancer, as enhancer malfunctions are central for pathogenesis of cancer. And the more we learn about function of factors that are involved in regulation of enhancer function could help us develop targeted therapeutics for treatment of cancer. So what we have learned during the past 20 years in my lab, during the past 20 years in my laboratory, as shown on the left-hand side, is that studying of the hematological malignancies in infant leukemia, the infantile leukemia, has provided us with information for the treatment of adult leukemia, and I'm going to describe that to you during my talk. So, as Steve mentioned, as Charlie followed through, is that stem cells are very central to cancer pathogenesis. The cancer stem cells have been described for many years, and what we know, the division of these cancer stem cells to many progeny cells and unregulation of the process is at the heart of cancer. But at the heart of the stem cells exists our chromatin in the center of the nucleus. And the function of the chromatin is to provide the genomic information, highly packaged. And as you heard from Steve and Charlie, chromatin play a very important role in packaging this very long-necked DNA within our chromosomes. And the packaging of DNA and unpackaging is an important process during the developmental regulation. What we have learned is that chromatin, as shown as the little balls over here, I don't know if the arrow, there we go, the little balls in here are involved in packaging of the DNA. And as you heard from Steve, DNA itself is post-translationally modified, and the chromatin can be moved around, as Charlie mentioned, with factors such as switch-SNF and remodelers moved around. Also, in addition to the modification of DNA and movement of the nucleosome itself, the tail of nucleosomes are also modified and called histone modification, the post-translational modification. And these modifications globally, altogether, DNA modification, movement of nucleosomes, and histone modification play a central role in packaging of our chromosome. And during the time of need, when genes need to be expressed, these regions are either modified, moved for open room for the basal machinery to recognize the sites and initiate transcription. And malfunction of any factors involved in this process, albeit being DNA methylation, movement of nucleosome around DNA template, or histone modification, have been shown to involve in pathogenesis of not only cancer, but a large number of human disease as well. So I'm showing to you in here the chromosomal spread in human cells, and these are from patients that bear translocations within a gene named mixed lineage leukemia, or stand for MLL. And over 30 years ago, Janet Rowley here in the city of Chicago demonstrated that chromosomal translocations, specifically in the MLL gene, are associated with pathogenesis of hematological malignancies. At the time of its proposal back in mid-70s, it was considered to be a heresy because translocations are not what's supposed to give us cancer, but it were the oncogenes and tumor suppressor genes. And it was shown in subsequent years that translocations indeed are involved in pathogenesis of hematological malignancies. And clinicians in children's hospitals all across the world can take a look at such cytogenetics and start developing therapeutic pathways for treatment of different forms of translocations that are involved in leukemia. On the left-hand side, I'm showing you a translocation on chromosome 4 into chromosome 11. So you can see in this chromosomal spread, there is a translocation between the armor portion of the chromosome 4 into 11. On the right-hand side, I'm showing you two translocations. One is a child with 1119 translocations into the ELL gene, another child into the 1119 translocations into the ENL gene. And in the bottom, you can see basically the long line MLL is the gene of about 4,000 amino acids, and two of its partners, ELL and ENL, have been shown in this figure. And the translocations of MLL to ELL, MLL to ENL, and other partners do indeed result in pathogenesis of cancer, as work by Terry Rabbits and colleagues showed if you knock in these factors into the endogenous location of MLL, the animal do indeed develop leukemia. So the translocation are central in the process. And my laboratory has spent a good bit of time during the past 20 years trying to figure out what is the commonality of these translocation partners in leukemic pathogenesis. And we have learned that many of the MLL translocation partners are actually found within a complex regulating the elongation stage of transcription that has to do linked to the elongation control, transcriptional elongation, checkpoint control, and leukemic pathogenesis. But alongside studying these from the patient samples, and they're shown mostly are newborn all the way to early adolescence on how they find into leukemia, we also wanted to define how MLL itself functions in this process. But before we move on that, let me describe the disease to you. So leukemia is basically hyperproliferation of our white blood cells in here. You can see on the right-hand side, you and I will have about 1% white blood cells in our total cells analyzed. On the left-hand side, we can look at some of these patients right over here, 80% white blood cell in this child. And many of these kids die from infiltration of these white blood cells to major organs, including liver, kidney, brain, and there's a lot of problem with the infiltration of these cells in these major tissues, which causes the complication. So the major question is that why translocation of MLL to many partners result in pathogenesis of leukemia, hyperproliferation of these white blood cells? And the question that we had many years ago was that what is the function of MLL itself? And we took advantage of multiple model systems in yeast, Saccharomyces cerevisiae, using genetic and biochemistry to understand the role of MLL homolog in cerevisiae, and moved on to metazoans such as Drosophila melanogaster, which provides fantastic genetic tools to study the role of these factors in regulation of gene expression, and then on to a mouse model system, and we're rechecking distinct from patient samples who have these leukemias. So the main question is that what is the homolog of MLL in the mammalian cells? Because we spent several years trying to purify MLL from the mammalian cells, and we failed miserably because we were not able to find MLL at its right side. And that was somewhere around about 400,000 Dalton on SDS page. Later, we learned from work of Stan Korsmeyer that MLL is actually clipped by an enzyme named taspase right over in the cleavage site in here, and this cleavage results in holding of MLL together with the 200,000 Dalton polypeptides, which are involved in the functionality of the protein. But we didn't know that at the time, and we decided to move on to an organism where we could do a lot more biochemistry and genetic, and we identified SET1 from Saccharomyces cerevisiae as the MLL homolog, and we start purification of SET1 from about 300 liters of yeast extract, and we start from the top of the chart over here on the left-hand side and pass these extracts over all these columns, and we purify the complex we name COMPASS. COMPASS stands for complex proteins associated with SET1, and what we showed that COMPASS, in the presence of S-adenosylmethionine, can mono, di, and trimethylate its nucleosomal substrate. For COMPASS was the first histone H3K4 methylase isolated, and we characterized it in yeast, Saccharomyces cerevisiae, and we showed that is an H3K4 methylase. In subsequent year work from my laboratory showed that there is only one COMPASS in Saccharomyces cerevisiae, shown on the top. The green subunits that are shown, CPS 40, 60, 25, 50, and 30, are regulatory subunits, and they're very important for the COMPASS function. So if you express that one by itself, is dead as a doornail, but it functions within its complex. And COMPASS is a lot like polycomb, because polycomb also requires this function within its complex, the PRC2 complex. So we followed through from our yeast studies into Drosophila melanogaster, and we showed that in the middle there are three COMPASS family members in Drosophila melanogaster: SET1 COMPASS on the left-hand side, the trithorax COMPASS on the on the middle, and then TRR or trithorax-related COMPASS on the right-hand side. Again, similar to yeast, the green subunits that are shown over here and the Drosophila are very much regulatory of the COMPASS function. SET1, trithorax, and TRR all are K4 trimethylases in melanogaster. We carried through from Drosophila all the way into the mammalian system and purified compass family members both from mouse and human cells, and we showed that set 1 is divided into set 1A and B compass, trithorax is divided into MLL1 and 2 compass, and TRR is divided into MLL3 and MLL4 compass. The most exciting part was that to show that the K4 methylation function of MLLs, or the compass family members, is non-redundant. For example, if we delete, or the community have done also as well, set 1 or set 1B, or MLL1, or MLL2, or MLL3, or MLL4, this results in embryonic lethality, indicating that their function is non-redundant. And our biochemical studies demonstrated that there is a division of labor among K4 methylation within the compass family. Most importantly, we showed that the MLL3, MLL4 compass are histone monomethylases, and they only function on enhancers, the 98% of the chromatin or genome that is involved in regulation of gene expression. So as this work was going on in our laboratory in defining the role of MLL3-4 compass as a histone monomethylases and regulation of enhancer function, a fantastic study came out from Eric Lander's group in the study of all the sequencing of the large number of genomes from different forms of cancer, solid or hematological malignancies, and they find out of about 5,000 tumor analyzed, handful of genes that is shown in here in the right-hand side of the corner are involved or mutated in large number of cancers. P53 sits on the top right over here. To our surprise, we found out that 3 members of the MLL3-4 compass, MLL3, MLL4, and UTX, are all found mutated in large number of cancer. In a sense, nature has done the genetic for us in here. Similar to complex that Charlie described to switch NIF, mutations of MLL3 and MLL4 are also associated in pathogenesis of large forms of cancer. So the question becomes, why is it that a point mutation in a general transcription factor, such as MLL3 and MLL4, results in pathogenesis of a tissue-specific cancer? And we think that enhancer malfunction clearly addressed that question. So if you take a look on the top panel in here, so let's assume that this cell in here is a pancreatic cell, and there are pancreatic-specific transcription factor that can bind to pancreatic-specific enhancer and involve in recruitment on the bottom factors such as MLL3-4, ELL3, and other factors. Recently we published the Zik family members to these enhancers. And once all of these factors are recruited to enhancers, through some magical thing that we still don't know, the enhancers are looped over into the promoter and regulate the expression of a given gene. So now assume that we have a mutation in the MLL3-4 at the site that it binds to its transcription factor in the pancreatic cells, and this mutation could result in locking in mechanism of the MLL3-4 binding to that transcription factor, and it'll never let go. So you get constitutive expression of a given oncogene only in the pancreatic cells, resulting into pancreatic cancer. So we think that enhancer malfunction describes why point mutation in a given transcription factor is associated with tissue-specific cancer, is because happening through transcription through enhancer regulation. If this is true, we would want to move on and develop compass inhibitors for compass or enhancer therapy, and I don't have time to get to the details of the studies, but recent work from my laboratory has identified that there are inhibitors of compass function, both the stability or regulation of its catalytic properties. These inhibitors can be used for the treatment of leukemia, at least in the mouse and in the cells that are developed. And hopefully once everything is in a row, we're going to move on to using these drugs in a phase 1 trial studies to develop compass inhibitor or enhancer studies in here. With that, I stop and thank a fantastic group of colleagues from my laboratory, whose picture is shown over here. Compass is so popular in Chicago that there's a bar named Compass Bar, and we regularly meet there for our discussions. With that, I stop and answer your questions.
Okay. Great. Thank you, Dr. Shilatifard. Thanks also to Dr. Baylin and Dr. Roberts for your very engaging presentations. We're going to move along quickly now to some of the questions that have been submitted by our online viewers. As a quick reminder to those of you watching us live, you can still submit your questions by clicking the Ask a Question button that's below the slide window. typing your question into the message box and then clicking OK. So the first question I wanted to ask here, so someone sent in a question specifically asking Dr. Roberts about whether the loss of SNF5 affects different cell types differently, whether loss of SNF5, whether the ability of SNF5 loss to promote oncogenesis depends on the tissue type or the cell type that loses the gene encoding SNF5. But I guess this question could also apply to MLL, which Dr. Shilatifard talked about. Why do mutations in MLL specifically or disproportionately affect white blood cells, leading to leukemia? So maybe Dr. Roberts, if you could address tissue or cell type specificity of SNF5 loss first.
Yeah, I think it's exactly what Ali was saying. We see an exquisitely specific dependence upon cell type that is driven by these cancers and these genetic loss. The epigenetic landscape has to be receptive, so it's only certain cell types, only certain epigenetic landscapes in which loss of these will drive cancer onset. And I think it's very similar to what Ali was talking about, that is an interaction with transcription factors that are dictating cell fate and perturbation of the ability of those transcription factors to execute their differentiation program. Loss of that ability is what is then contributing to the predisposition. Ali?
Yeah, I fully agree with Charlie, and I think I definitely think that genetic was shown in Drosophila as a function of trithorax, and remember, the Compass family are part of the trithorax families, and what Charlie talked about on switch SNF. I bet you a lot of these things are functioning together on enhancers. And I think this is something that the field has not really looked upon, but I think developing great antibodies against component of Compass and switch SNF and then going and looking at their function on enhancers from patient samples and from normal cells, do we get more information on how enhancer malfunction could involve in bringing Compass and switch SNF together to result in this cancer. So my thought is that enhancers would be the place that I would look first.
Okay. And so now I want to ask another question that's also sort of about specificity, but about specificity of the enzyme. So I know that, for example, histone deacetylases, despite the fact that they're called histone deacetylases, also have deacetylase activity towards other proteins in the cell. So can some of these components of some of these chromatin modifying complexes that have enzymatic activity towards other targets also be affecting oncogenesis in cells that bear mutations in these genes?
So I can move forward.
Yeah.
So I fully agree with you. Actually, as a field, we got together and we proposed a new nomenclature for many of these epigenetic functions. And instead of calling them a histone methylase or histone demethylase or histone acetyltransferases, we renamed them as lysine methylases or lysine demethylases or lysine acetylases because they do have a lot of substrate that are non-histone. And I think only focusing on histone substrates probably could lead us into wrong path for many of these factors. But more importantly, in addition to their catalytic function, our unpublished works really points towards the context dependency of these enzymes, that they do have function that are independent of their methylase or demethylase or acetyltransferase functions, because these are large protein, 400,000 Dalton protein. It can't be just only a function of methylation, but it provides a context for recruitment of other factors, and these protein-protein interactions and many of these loci in the genome are just or perhaps more important than the catalytic properties. But as a field, we haven't looked into it because there is not an easy way of doing so.
Let me ask one final question. We've gotten a lot of questions and some really good questions, but I'm going to ask one last one, and that's about personalization. So, every tumor is different, and even within the same tumor, the cells can change over time. So is there a way, or how can you personalize epigenetic therapy for specific tumors?
I think, that's Charlie, I think we really need to be able to go at the higher levels of the chromatin. I think within the switch-NIF mutant cancers, and even within tumors that seem to be the same type, the downstream pathways are not identical. And again, it's just as Ali was saying, it's dictated by what transcription factors are there. And so there may not be such a key downstream target that underlies all of these cancers, but rather context dependence. And so if we're going to try and be able to more broadly target them, we're going to have to go right at the regulatory mechanisms that are being perturbed in the first place, and that's at the level of chromatin regulation or DNA methylation alteration.
And this is Dr. Baylin. I gave a brief example of a small subset of patients with the world's biggest killer, non-small cell lung cancer. Only a few percent that respond to epigenetic therapy, the type we were giving them alone. But if we could identify the pathways altered that counted for that, this would be thousands and thousands of people around the world. So I think it's the pathways like you're hearing. You have to know when you target a given mutation or a given epigenetic change or target, you've got to look at the pathways that are modified and really cull down who are they signaling to and what are the events surrounding them.
Mm-hmm. Mm-hmm. Well, I think we are out of time for the webinar, so I think we need to wrap it up. So I want to first thank Dr. Baylin from the Johns Hopkins School of Medicine, Dr. Roberts from the Dana-Farber Cancer Institute, and Dr. Shilatifard from the Feinberg School of Medicine for being with us today and for their fascinating talks and for answering our questions. Also, I'd like to thank our online audience for the questions you submitted to the panel. I'm sorry that we didn't have time to get through all of them. We got a lot, and we really appreciate your participation. Please go to the URL that's shown now at the bottom of your slide viewer to learn more about resources that are related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This webinar will be made available to view again as an on-demand presentation in approximately 48 hours from now. We'd love to hear what you thought about the webinar. You can send us an email to the address that's now shown on your screen, webinar@AAAS.org. Again, thank you to our panelists and to Cell Signaling Technology and the Koch Institute for their kind sponsorship of today's seminar. Thank you all for listening. Goodbye.